Preparation model paper 5, ASRB 2024 preparation. This is especially related to plant virology. So I am N. H. Shankar Reddy, PhD plant pathology. The first one is mother of virology and serology. So we might have studied like father of plant pathology, father of virology, father of bacteriology, and various other kinds of things. Majorly we used to see only fathers. There is no mother, right? But when it comes to here, mother of virology, there is a person called mother of virology and serology. Helen Alice Purdy-Beale, Rachel Carson, Mary Curie, Grader. So generally this Helen Alice Purdy-Beale is generally considered as mother of plant virology. The Rachel Carson, she wrote a book called Silent Spring, right? That was published in 1962. That is also a very important book to initiate environmental revolution. So generally, the answer is the mother of virology, Helen Alice Purdy-Beale. Mary Curie is not at all related to our field. And the second one is the largest viral host range having the pathogen. Which pathogen is having the largest viral host range? Tobacco mosaic virus, tomato mosaic virus, cauliflower mosaic virus, none of the above. Generally, tobacco mosaic virus having the largest host range. So we do consider this as the model organism for plant virology. So the largest host range is having by tobacco mosaic virus. TMV produces amorphous or crystalline bodies are known as. Generally, if it is a viral infection, they will produce some kind of identification structures. We can generally call it as inclusion bodies. So each and every virus produces different shapes of inclusion bodies. The TMV producing crystalline or amorphous bodies are is known as X bodies. Of course, we do have a detailed explanation about this different kinds of inclusion bodies that is produced by the plant viruses. So the answer is TMV producing X bodies are known as X bodies. Okay. Onion Irish yellow spot is actually transmitted by. They're asking transmitted by. So it is known to be transmitted by thrips. Banana streak virus is transmitted by. Banana streak virus is transmitted by mealybug. Very important one. Not only banana streak virus, there is one more virus. I'll show you. Yes, coconut swollen shoot virus, banana streak virus. These two viruses are known to be transmitted by an insect vector called mealybug. Both the mealybug scientific name is Pseudococcus nijalensis. The next one is the sixth one. Who first demonstrated the cross inoculation of tobacco mosaic virus? So the cross inoculation of tobacco mosaic virus was first performed by Clinton, George Clinton. Seventh one, who first discovered the foot and the mouth disease of virus and forms of other animals. Generally, it's a Paul Froch in 1898. He was the first who discovered this foot and mouth disease. The first DNA virus that is completely genome sequenced. First RNA virus means actually generally if you ask me first virus to be completely genome sequenced, it is a cauliflower mosaic virus which was sequenced in the year 1980. First RNA virus to be completely genome sequenced is tobacco mosaic virus 1982 by Golet et al. So if I ask you like what is the first virus to be completely genome sequenced, since they have not mentioned whether it is a DNA or RNA, the credit goes to cauliflower mosaic virus because the cauliflower mosaic was sequenced in 1980 itself, whereas the TMV was sequenced in 82. So the first sequencing plant virus is cauliflower mosaic virus only, right? So they are asking first completely genome sequenced virus and sequenced by, they are asking. Simply in 1980 cauliflower mosaic virus was sequenced by Frank et al. Even though if you remember date year also, we can easily answer because Frank et al. Sequenced in 1980. So these are all the couple of important genome sequencing. First fungus completely genome sequenced is Magnoporthegricia, sequenced by Dean et al. Year 2005. First bacteria completely genome sequenced is Xylella fastidiosa, A.J.G. Simpson and 2000. First DNA virus completely genome sequenced is cauliflower mosaic virus, sequenced by Frank et al. Year 1980. First RNA virus completely genome sequenced is tobacco mosaic virus, Golet et al. 1982. First viroid is potato to spindle tuber viroid that was sequenced by Gross and Dixon 1978 and 79. Yeast was sequenced by 1996. Saccharomyces cerevisiae. Okay, so nematode is first nematode sequenced is Chinorhabdis elegans that was sequenced by Andrew Fire and Craig Mello. These are all the two persons who discovered this RNA interference. We can also call it as post-transcriptional gene silencing or quelling various kinds of other terminologies are there, right? Can also called as RNA interference, right? That was first identified in a nematode called Chinorhabdis elegans, and it is also known to be the first plant parasitic nematode to be complete, not sorry, first parasitic nematode to be completely genome sequenced. Next one is the red bread mold, that is Neurospora crassa. It was sequenced by Gallagher in 2003 et al. On the right side, I have also given the genome sequencing size. So size is not necessary, but you remember the agent or pathogen, the first pathogen and the scientist involved and the year. Sometimes we do expect years as well. Okay. And 7th and 8th we have already discussed. Anyway, I'm going to 9. Virus moves from cell to cell through. So generally viral movement from one cell to another cell is mediated by plasmodesmata. For example, this is one cell, this is another cell. Actually, these two cells are actually connected by a bridge-like structure called plasmodesmata. Generally, the virus is bigger when compared to plasmodesmata. Plasmodesmata is smaller than that of plant virus, but still this plant virus is moving through because it produces a protein called movement protein. So with the help of movement protein, the virus is mediating their movement through this plasmodesmata, so that it will be very easy to go from one cell to another cell through a pipe like structure or through a channel like structure. Again, if the virus want to come outside, again through pinocytosis, again it need to enter inside. It will be a very big process, so that it is a very easy if it is transmitted through plasmodesmata. Generally majority of the viruses, all viruses are known to be transmitted from cell to cell through plasmodesmata. In fact, not only plant viruses but also viroids through plasmodesmata and cell division. Okay, hypersensitive response was first seen in which crop? So the hypersensitive response was for the first time it was discovered in a crop called tobacco. In tobacco only hypersensitive response was first discovered, right? Next one, elicitors of tobacco mosaic virus. So tobacco mosaic virus elicitor is known as coat protein. The coat protein, for example, this is a nucleic acid. It contains a protein coat or coat protein. The coat protein itself is considered as elicitor, which actually elicits a defense response. Okay. First satellite virus was reported by. There are a couple of first reports are there. It will be very important. Like we do expect a couple of questions from that. So generally, this first satellite virus was first discovered by Cassanis in 1962, and there are other also there that I'm going to tell you in the next slide. The nucleic acid is the infectious material of plant virus. Generally, nucleic acid, plant virus contains two components. One is the protein coat. Second one is nucleic acid. This is the nucleic acid. This is the protein coat. Generally, before W.M. Stanley, who is generally considered as the first person who sequenced who actually crystallized the tobacco mosaic virus in a pure form, he thought that protein coat might be the infectious material. But the later, there are two scientists called Geiger and Scram in 1956. This question was asked in two times. If you recall the previous year question papers, in fact, we have also published the last 10 years in question papers. Please take a look into the playlist. Anyway, coming to the point, so generally the nucleic acid is the infectious material of plant virus was given by Geiger and Scram in 1956. If you want, you can just take a look into the previous year question paper. You will get this question, right? So it is a very, very important. Before, Stanley thought that the protein coat is the infectious material, but later Geiger and Scram identified protein coat is just a protective material, but nucleic acid is the infectious material of plant virus was reported by Geiger and Scram. And these are all a couple of first reports. Since I have told you satellite virus was Cassanis, right? So first fungi to be reported by Anton Tiberry, lead blight of potato, Phytophthora infestans. First bacteria to be reported by T.J. Burrill, fire blight of apple, Irvinia amylovar. First virus to be completely genome sequenced is Beiser tobacco mosaic virus, TMV. First viroid is Diner and Raymer, potato spindle tuber viroid. First virucide is Randless, valvate tobacco mosaic tobacco model virus. Phytoplasma Doe et al, Mulberry dwarf. Spiroplasma Davis et al, constant. Satellite virus they have asked, Cassanis 1962, tobacco necrosis satellite virus, and this tobacco necrosis satellite virus is also known to be the smallest plant virus with 17 nanometer in diameter. Mycophages Hollings 1962, Cyanophages Saframan and Morris, Bacteriophages Twart and Harrelly. The term bacteriophage was given by Harrelly, and the term bacteriophage means bacterial eater. Bacteria phase means eater, bacterial eater. So this is how it will work. Next one is in virus, infected leaf or virus concentration is more in where the virus concentration is more in, especially the areas of chlorotic areas where the virus inoculum load will be more. Potato leaf roll viruses of Gemini virus comes under the genome. So genome of potato leaf roll virus, they contains a genome of single stranded DNA. Okay, all the Gemini viruses, not only potato leaf roll virus, all the Gemini viruses, they contain genome as single stranded DNA. Viruses are, next question, viruses are made up of nucleic acid and protein, and this nucleoprotein nature of viruses was for the first time it was demonstrated by Bowden and Pyrie. So they actually coined nucleoprotein. Generally virus is called as nucleoprotein because virus contains a nucleic acid and as well as a protein, both. That was first reported by Bowden and Pyrie. So they named it as a virus is a nucleoprotein, which means a combination of nucleic acid and protein. Okay, so the next one is PCR can amplify by PCR can amplify both the DNA and RNA. Of course there is a couple of crosstalk is also that it can only amplify DNA, it can only amplify RNA. There is no PCRs can amplify RNA, such kind of things will be there, but there are PCRs available that can sequence both DNA and RNA as well. Sugarcane viruses is transmitted by, it is known to be transmitted by a Perxneella saccharida. Okay, next, indicator plant for citrus exocortosis. Of course there are a couple of indicator plants that we are using in now plant virology. Then you can ask me, what is indicator plant? It is a plant which is used to identify the presence or absence of, like, a indication or identification, right? So the use of indicator plant in plant virology was first thought by K.M. Smith, probably in 1930s or 31, I think so. So here there are a couple of indicator plants like citrus exocortosis for all viruses generally, and the indicator plants that I have already disclosed in the later slides. Now we'll see the indicator plant for citrus exocortosis is this trifoliate orange. Trifoliate orange is considered as a citrus exocortosis indicator plant. The size of bean common mosaic virus. Bean common mosaic virus size ranges from 12 into 750. Now we will see the sizes of other couple of plant viruses. So satellite tobacco necrosis virus, and I have already told you that was first discovered by Kasani. So satellite tobacco necrosis virus, it is the smallest plant virus, which consists of 17 nanometer, and tobacco necrosis virus 26, brome mosaic virus 28, copy mosaic virus 28, tobacco ring spot virus 28, Humor mosaic virus 30, Waika virus 30, tobacco bushy stunt virus 30 to 35, alfalfa mosaic virus 30 to 57, cauliflower mosaic virus 50 to 52. Lettuce big bean virus 60, potato yellow dwarf virus 90, Rio virus 65-70, wound tumor virus 70, sugarcane mosaic virus 11 into 750, bean common mosaic virus 12 into 750, plumpox virus 12 into 760, Badna virus 300 into 20, tomato spotted wilt virus is 100 nanometer in shape, sizes, which are spherical in shape. So these are all the couple of important plant viruses and their sizes. The oldest known plant viral diseases. The oldest known plant viral disease is tulip color breaking. That was first reported in 16th century probably. So since even though it's a viral disease, but the economy of the crop actually goes high because of tulip color variations, or petal changes to different colors. So it actually gives more price than expected. So tulip color breaking, that was actually reported in 16th century, around the time of 16th century. The next one is the oldest plant viral disease was recorded in the year, as I told you, 16th century. Okay, founder of plant virology is considered founder of plant virology. Generally, Bijernik is considered as a founder of plant virology because he is the first one who proved that tobacco mosaic disease was caused by virus, and coined the term virus, and various other things are done by Bijernik only. That's why he is considered as the father and founder of plant virology. Next one, who demonstrated that strains of TM and potato virus X can be distinguished serologically. So this both the potato virus X, PVX and TMV, that can be distinguished by using serologically was first demonstrated by Chester. Name the scientist who applied X-ray analysis to purify the preparation of tobacco mosaic virus. That was Bernal and Frankochin. The first serological classification of plant viruses was done by who first tried to serologically classify the plant viruses. So it was first done by Chester. The bimodal transmission was the first proposed way. What is bimodal transmission? Bimodal transmission means generally the transmission of plant virus by means of two agents. One is, for example, you can take an example of the cauliflower mosaic virus. It is known to be transmitted by aphids and as well as sap transmission also. And it is the first reported virus to be reported as a bimodal transmission by Chaffant and Chapman in 1962. That is a cauliflower mosaic virus. The first bimodal transmission was reported in the virus cauliflower mosaic virus, that was first reported by Chaffant and Chapman in 1962. And I will tell you one more important point about this cauliflower mosaic virus, and it is also known to be the first virus or the first DNA virus that is completely genome sequenced, and is also known to be the first virus which contains double stranded DNA as a genome. So this is the first virus where the double stranded DNA contains genome, and it is also first reported virus whose genome contains double stranded DNA, and also first bimodal transmission reported plant virus as well. And anyway, next one, PGNP sterility mosaic virus was the first reported from. It was first reported from which state they are asking? It reported from Bihar. Cotton leaf curl virus is transmitted by. Cotton leaf curl virus is transmitted by whiteflies, right? Handbook on phytopathogenic trees was written by. It was written by Holmes. Local lesion essay we might have studied earlier, like local lesion essay was given by Holmes. So he also wrote a book called Handbook of Phytopathogenic Viruses. Who reported the use of carborundum powder, which increases the efficacy of local lesion assay. So this carborundum powder, chemically known as silicon carbide, which is used in local lesion assay. Then what is the purpose? So the purpose is generally for making the wounds, or to make some kind of disturbances on the leaf surface, so that the virus sap can easily penetrate or enters into the plant cells. So that is main purpose. The carborundum powder generally actually increases the efficiency by making the wounds. That is the only thing, nothing else. And anyway, the use of carborundum powder which increased the efficacy was first demonstrated by probably I think Rowland and Tompkins, I think so. A virus family always ends with virus family. It is always ends with I think Viridae. I think I'll show you a couple of other things. Take a look at this. Order always ends with the virals. Family always ends with the Viridae. Subfamily always ends with the Viridae. Genus always ends with the virus. So since they've asked family, family always ends with the Viridae, right? Next one. Number of viroid species are discovered so far. So far there are 40 viroids are discovered. All the plant viroids, all the viroids are known to infect only plants. There is no other viroids are reported that infect humans or some other things. It is known to infect only plants. There are 40 viroids are discovered. All the viroids contains a single stranded RNA as a genetic material, and all are known to be transmitted by sap transmission, or we can also say mechanical transmission. Sesame phyllodes was first time reported by. It was first time reported by Pushkarnath. Infectious and non-infectious RNA was purified by TYMV by. Who purified it? They are asking. So probably it is a Markham and Smith. Double-stranded DNA in cauliflower mosaic was first time reported by. It was first time reported by Shifford et al. In 1968 probably. Lambda pi or pi x174 was actually discovered by. It was discovered by Friedrich Sanger, and he got 2 Nobel Prizes. Okay, tadpole shaped pathogen. Which of the following is a tadpole shaped pathogen? Bacteriophages. Bacteriophages look like actually a tadpole with the 6 legs, or tail fibers we would say, right? So the first indication on the shape of plant viruses was given by Takashi and Rollins. Aphid transmission of plant viruses was first time reported by. It was first time reported by Hogan et al. Next one, who emphasizes the importance of a nucleic acid in viral replication? It was Hershey and Chase who actually put a thing forward that nucleic acid is the major factor in viral replication. Before it is thought that protein code plays a major role, that is the infectious factor, right? So after this Hershey and Chase experiment, they came to a conclusion that this nucleic acid is a very important factor in viral replication. The next one, the apical shoot meristem culture technique was used to produce a virus-free stocks, and dahlia and potato were the first time reported by. It was first time reported by Morrell and Martin in 1952. And this is generally a tissue culture. Okay. The scientist who describes a technique for propagation and infecting protoplast with viruses, that was Takabe and Oshuke, that was the technique was given by. The use of a coat protein gene to produce the virus resistant transgenic plants was first described in a TMV by. That was first described in TMV by Paul Abel et al. It is a very important one. Coat protein mediated gene resistance or coat protein mediated resistance was first given by Paul Abel et al. Powell and Abel et al. 19 probably 86, I think so, I'm not sure. Mycoviruses was first reported by I think in the previous slides and the second slide or maybe in third slide we might have a very a tabular column, right? Pathogens for their first discoveries and all those things that was given, right? Mycoviruses are the viruses that infect fungi. So that was those are all called as mycoviruses. That was first reported by I think Hollings, right? Wound tumor virus, generally the size of wound tumor virus is 70 nanometer in diameter, which contains double-stranded RNA as a genetic material. So this wound tumor contains double-stranded RNA as a genetic material was first given by, they're asking, it was given by Black and Markham. Virus transmission through nematodes was first demonstrated, but it is a very important one. Hewitt et al. Probably in 1958, I think so, is the first one who demonstrated that virus can transmit through nematodes. Wound tumor was alerts are right. Virus transmission through nematodes was first reported by Hewitt et al. Okay. ICTV, International Committee on Taxonomy of Virus, was established in the year 1966, whereas the same ICTV name changed to just a minute. ICNV, the year establishment is 1966. The same ICNV name changed to ICTV in the year of 1973. Since they have asked ICTV, the year is 1973. The ICTV came from ICNV, International Committee on Nomenclature of Virus, which was established in 1966. The name ICNV changed to ICTV in the year of 1973. What is ICNV? Just now I told you, International Committee on Taxonomy of Viruses. I think the answer is A. The next one is the first report of cyanophases was done by Safferman and Morris in 1963. Okay, so virus that infect blue-green algae is known as cyanophases. That was reported by Safferman and Morris, LPP1, right? The next one is sensitivity of MLOs, mycoplasma-like organism, to tetracycline antibiotic was first reported by Ishii et al. The term virus means enzyme, poison, toxin. So generally it comes to poison. The term virus means the term virus is poison or venom or slimy liquid. It was given by M.J. Bijanic, generally we used to consider the father of plant virology, right? So the term virus means poison or slimy liquid or venom. Next one, which scientist is associated with the discoveries that actually concerning the control of enzymes and viral synthesis? So enzymes and viral synthesis they are asking. So it was Andrew Love. He was the first person who actually discovered the the genetic control of enzymes and the synthesis of virus, how it is actually happening and all those things. That was given by Andrew Love. He is also a Nobel laureate. In virus taxonomy, order always ends with the virals. In mycology, order always ends with the ales. When it comes to here in virus taxonomy, order always ends with the virals. Enzyme required for the replication of virus. So in the, if you might have studied the general characters or general properties of plant viruses, there itself we might have studied for the replication of plant viruses, it requires RNA polymerase 1. For the replication of viroids, it requires RNA polymerase 2. So the answer is RNA polymerase 1, since they have asked about virus. Tests used for the identification of viroids. There is a test called page test. So this test is generally recommended for the identification of this viroids. I think polyacrylamide gel electrophoresis, right? And India's first reported viroid. What is India's first reported viroid? This is a tomato bunch top viroid is known to be the India's first reported viroid. Shape of TMV, tobacco mosaic virus. Tobacco mosaic virus is generally rod shaped. This is exactly look like a rod shaped, where this capsid is arranged like this. So the size size will be around 18 into 300 nanometer. Jacob disease in human is mainly due to caused by prions. So what is prion? Prion is a infectious protein, I would say malfunctioned protein. So the malfunctioned proteins are generally known as prions. So prions causes general couple of diseases. In humans it causes Jacob's disease, Kuru disease, some other kinds of diseases are there. So Jacob disease is mainly due to caused by prions. Okay, in general the amount of nucleic acid present in plant viruses ranges from 5 to 40% of nucleic acid and 60 to 95% of protein code. That is a general range of all plant viruses. The next one, in general, the amount of nucleic acid, generally 5 to 40% we have discussed, right? And in general, the amount of protein code, that also we have discussed, generally it is 60 to 95%. The next one is a genome of mycoviruses and cryptoviruses they're asking. So genome of mycoviruses and cryptoviruses contains double-stranded RNA. Whereas double-stranded DNA, one example is a cauliflower mosaic virus. Since this mycoviruses and cryptoviruses contains double-stranded RNA, okay. The amount of protein in TMV contains 95%, but there is an exception. Generally, all plant viruses contains 5 to 40% nucleic acid and 60 to 95% protein code, but there is an exception called TMV. TMV contains 5% nucleic acid and 95% protein or protein code, we can also say, right? So the next one is the phytoplasma belonging to the order Acheloplasmatales, whereas spiroplasma belonging to the order Entomoplasmatales. So I think the next question is same in Phytoplasma Acheloplasmatales, Spiroplasma Entoplasmatales. Technique used for the detection of phytoplasma. So there is a technique called DAPI technique, and also Dines strain is also used. So probably the answer is both A and B, DAPI and Dines strain. So this generally while we are staining phytoplasma, when we stain this DAPI, I think we do get a color called blue color, I think so. And this color also sometimes it was asked, okay. So the technique used for the detection of phytoplasma is a DAPI technique or DAPI technique, and also Dines strain is also used. And these are all the couple of nucleic acid content of plant viruses. All viruses generally contains 5 to 40% and the protein code is 60 to 95%. Tobacco mosaic virus contains 5% protein or nucleic acid and 95% protein code. And a turnip yellow mosaic virus contains 34% nucleic acid and 66% protein code. Potato virus X and virus Y contains 6% nucleic acid and 94% protein code. Copy mosaic virus 33 nucleic acid and 67 to 69%. Cucumber mosaic virus 18% nucleic acid and 82% protein code, whereas the tobacco ring spot virus contains 40% nucleic acid and 60% protein code. These are all a couple of nucleic acid as well as protein compositions. Next one, the most important vector in transmitting a plant viral diseases, and there are a couple of various vectors are that among the majority of the plant viral diseases are known to be transmitted by an insect vector called aphid. Around 50 to 60% alone are transmitting through this aphid only, okay. Little leaf of brinjal is transmitted by leaf hopper Hishimonas spicetus, whereas sesame phyllode is also known to be transmitted by leaf hopper but voracious albicinctus. Sesame phyllode is voracious albicinctus, whereas little leaf of brinjal is Hishimonas spicetus. Both are leaf hoppers only, but the scientific name I am telling, okay. And one more thing is majority of the phytoplasmal and spiroplasmal diseases are known to be transmitted by leaf hopper only. Majority of the phytoplasmal and spiroplasmal, very few lightly one or two diseases may varies, but majority of the phytoplasma and spiroplasma are known to be transmitted by an insect vector called leaf hopper. The next one, coconut lethal yellowing is transmitted by plant hopper, Mundus crudus, Apalec, both. This coconut lethal yellowing known to be transmitted by plant hopper, and there are two insect species are there, Mundus crudus and Hepalexis crudus, both A and B. Next one, culture colonies with the fried egg or poached egg can be the characteristic feature of. So the characteristic feature of spiroplasma during the culture, this spiroplasma colonies are appeared as a fried egg or poached egg. When you are frying the egg or when you are poaching the egg on the pan, so it exactly look like a fried egg or poached egg. So the same spiroplasmal colonies also look like a fried egg or poached egg. The next one is which spiroplasma can be cultured by Saligo in 1937 actually for the first time. So I think for the first time it was cultured by Spiroplasma citri, I think so. The spiroplasma can be better controlled at what temperature? So when the temperature is exceeding beyond 37 degrees centigrade, the culture cannot come out, so that we can automatically control the culture, I am telling, okay. Citrus stubborn is a spiroplasmal disease are known to be transmitted by, it is known to be transmitted by leaf hopper Cercolephrus tenalis. These are all the list of phytoplasmal disease and their vectors. Little leaf of brinjal, leaf hopper Hishimonas spicetis, and we can also one more other vector called Amarcus pigutella, Sesame phyllodes, leaf hopper, Orosius albicindus, grassy shoot of sugarcane leaf hopper, Saccharosydney saccharivora, and lethal yellowing, coconut lethal yellowing, plant hopper Mendes crudus and Hapalexis crudus, rice yellow dwarf, leaf hopper Nephotitix virescens, mulberry dwarf, leaf hopper Hishimonas cellatus, European stone fruits, it is known to be transmitted by psyllids. As I told you, majority is known to be transmitted leaf hopper, but one or two are varies. One of such an example is European stone fruit yellosis, which is caused by known to be transmitted by psyllids. Next one is peach X disease, leaf hopper. Pear decline, pear silla, tomato big bud leaf hopper, sandal spike. So this sandal spike is actually the first reported phytoplasmal disease in India in 1966 by Verma. Okay, apple proliferation, that is known to be transmitted by vegetative propagation, and it is also the phytoplasmal disease. If you see a couple of preparation question papers, it is mentioned that apple proliferation is known to be caused by, or apple proliferation is caused by, it is known to be caused by phytoplasmal disease. Okay, phytoplasmal infection. The next one is apple proliferation. Yeah, just now we discussed it, right? Apple proliferation is caused by phytoplasma. The next one is European stone fruit yellosis, is a phytoplasmal disease that is known to be transmitted by psyllids. Okay, Sesame phyllodes is otherwise called as Sesame phyllodes is otherwise called as cephaloidy, otherwise called as stenosis, otherwise called as phyllomonia. So the answer is all the above. Okay, bacteriophages means bacteriophage. Phase means eater. Bacteriophage means bacterial eater. A virus that infects bacteria is known as bacteriophages. So the answer is A, virus infecting bacteria. La France disease, La France disease or La France disease is otherwise called as X disease, water stripe disease, dieback, all the above. It is all the above. It is a mycovirus. It is a first virus that was reported in mushrooms. Okay, this La France disease or La France disease, also known as X disease, also known as water stripe disease, also known as dieback of mushrooms. So the answer is all the above. Okay, cyanophages means, so phases means eater, cyanophages means blue green algae. Okay, right? So generally a virus that is parasitizing or infecting blue green algae is known as cyanophages. Answer is both. Answer is A. So the cyanophages was discovered by Safferman and Morris in 1963. If I ask you like who discovered this bacteriophages, you might aware of Twart and Herrelli. There are two scientists. Twart 1915, Herrelli in 1917. They individually discovered the bacteriophage. But the term bacteriophage, but the name bacteriophage was given by Herrelli in 1917. The cyanophages, Safferman and Morris, and the first cyanophage probably LPP1, Lingba, something as well, right? So the next one is virus cryptograms indicates. Cryptogram means short names. Okay, so generally virus properties using symbols is known as virus cryptograms. Why short symbols? Okay, who introduced the term plasmodesmata? The term plasmodesmata was first introduced by probably Strasburger, I think so. TMV amino acid molecular weight. The molecular weight of TMV amino acid is 17.6 kilodalton. Refractive index of TMV, tobacco mosaic virus. The refractive index of tobacco mosaic virus is 1.57. The next one is thermal inactivation point of TMV, tobacco mosaic virus. The thermal inactivation point is 93 degrees Celsius. In a couple of books it is mentioned as 90, but the general thermal inactivation point of tobacco mosaic virus is 93 degrees Celsius. But the majority of the plant viruses can be inactivated at the temperature range between 55 to 70, but there is a specialty TMV that can withstand up to 93 degrees Celsius. Okay, TMV genome can be altered with, so the genome of TMV can be altered with ADP, adenosine diphosphate. Okay, the number of ORF rings present in TMV. First of all, I will tell you what is ORF, open reading frame. So TMV contains 4 open reading frames. So the number of ORFs present in TMV is 4. Okay. The most widely used antibody in plant virology. In plant virology, we use an antibody called IgG. That is the most widely used antibody in plant virology. The next one is most commonly, again we have already saw it, right? Most commonly used antibody in plant virology is IgG. The next one is a viroid can replicate by, means they are asking viroids, what is the mechanism of replication of plant viroids? Generally, it is a rolling circle mechanism. It is a very important. Viroids can replicate by means of rolling circle mechanism. So binary fission we may do generally we do observe in bacteria, right? And enzyme used for the replication of plant viroid. I think this question we have already discussed earlier, but instead of viroid, it was their virus, right? So here generally for replication of virus we require RNA polymerase 1. For the replication of viroids we do require an enzyme called RNA polymerase 2. So the answer is RNA polymerase 2. CCR present in which group of viroids? First of all, we'll see what is CCR. CCR means a central conserved region. So the viroids are actually classified based on this CCR only, presence of CCR or absence of CCR. Generally, the CCR is present in the group of Avicennaviridea, whereas the Paspiviridea consists of no CCR, central conserved region. Okay, based on this only, these two groups of viroids are divided, Avicennaviridea and Paspiviridea. The next one is symmetrical rolling circle mechanism can be observed in. So symmetrical rolling circle mechanism can be observed in Avicennaviridea, whereas in Paspiviridea the mechanism is absent. In the next slide, I have placed like what is the difference between this Avicennaviridea and Paspiviridea, so that we'll have a better idea about this, right? So site of replication of Paspiviridae. So Paspiviridae generally replicate in a nucleus. Okay, number of nucleotides present in coconut kadang viroid. This is also one important one, like how many number of nucleotides are there. Each viroid contains and all those things. And the smallest viroid is coconut kadang viroid only, which consists of just 246 nucleotides, right? And it is known to be the smallest viroids reported so far. The next one is nucleotides present in potato spindle tuber viroid. So this potato spindle tuber viroid is known to be the world's first reported viroid by Diner and Reimer, right? So it consists of probably 359 nucleotides, 359 nucleotides. Yeah, here are the difference between Paspiviridae and Avicennaviridea. Generally, CCR is present in Paspiviridae. So anyway, actually in the previous MCQ I have discussed that CCR is present in Avicennaviridea, right? Anyway, please make it right. CCR is actually present in Paspiviridae, absent in Avicennaviridea. You can remember in this way also. Paspiviridae starts with P, present. Avicennaviridea starts with A absent. Like that you remember. And please change the answer for the previously discussed MCQ. CCR is actually present in Paspiviridae and absent in Avicennaviridea. Ribosomal activity in Paspiviridae, ribosomal activity is lack, and Avicennaviridea consists of ribosomal activity. The shape of the molecule is circular generally, and Avicennaviridea molecule is also circular. The secondary structure is rod-like, and when it comes to Avicennaviridea, it is a branched. Another important thing is rolling circle mechanism. Asymmetrical rolling circle mechanism is present in Paspiviridae, whereas a symmetrical rolling circle mechanism is present in Avicennaviridea. What is the site of replication when it comes to Paspiviridae? It is a nucleus. When it comes to Avicennaviridea, it is a chloroplast. It is a very important one. I think previously we have discussed it. Replication enzyme is DNA dependent RNA polymerase 2, and Avicennaviridea DNA dependent RNA polymerase. And the number of rolling circles, only one rolling circle mechanism can be observed in Paspiviridae, and two rolling circles. See, from this table also we do expect questions like just now we have CCR. They may also ask like what is the site of replication of Paspiviridae, what is the site of replication of Avicennaviridea, and the replication mechanism they may ask. Cleavage replication, host nuclear RNA when it comes to Paspiviridae, and hammerhead ribosome when it comes to Avicennaviridea. So the examples of viroids that comes under Paspiviridae is a potato spindle tuber viroid, coconut kadan viroid actually comes under this Paspiviridae. And there are a couple of viroids also comes under this Avicennaviridea, like avocado sunblotch, chrysanthemum chlorotic model. This is the largest viroid, chrysanthemum chlorotic model viroid, which consists of 401 nucleotides, in between 399 to 401. To be exact. Coconut kadan viroid is the smallest viroid, which consists of 246 nucleotides, right? Yes, these are all a couple of lists. If you see this, the families, only two families are there, Avicennaviridea and Paspiviridae. Avicennaviridea is a very small one, consists of avocado sunblotch, 246 to 251, chrysanthemum chlorotic model viroid, 398 to 401, and eggplant latent virus, 332 to 335, peach latent mosaic viride 335 to 329. So now in Avicenna viride, one thing is very important. We must remember there is a chrysanthemum chlorotic model viride. One is the largest viride so reported so far. And in past period chrysanthemum chlorotic stunt, citrus exocartos nucleotides important 368 to 375, and a couple of viruses are the potato spindle tuber viride 356 to 361. I think we might have discussed it 359, right? Like that. Tomato, this is not that much important. Coconut kadan, this coconut kadan viride is important. 246 to 247, and one more is also important. Coconut tinan gaja, which consists of 254 nucleotides. Other things like apple scare skin viride 320 into 334, grape vinylosis base builds. These are all a couple of virides. You just take a look into so that you will get like what are all the different kinds of virides are there. So this is general information about this classes of virides. And the next one is number of nucleotides present in citrus exocartosis. I think we have discussed it consists of around 362 to 371. So the answer is 371, right? What is the test used for the identification of BBTV? What is BBTV? Banana bunch top virus in Indian cassava virus. Actually, generally, what is the test used for the identification of banana bunch top virus and Indian cassava mosaic virus? Generally, tetrazolium test, very important one. What is the test used for the identification of banana bunch top virus? So tetrazolium test. There are a couple of tests are there which are actually used for the identification that I have placed in the next slide that will be useful for you guys. Okay, generally BBTV, tetrazolium chloride test. What is the in clastrovirus? Clastro means it generally a thread-like structure. Okay, virus which does not code any code protein. There is a one important virus, Umbra virus. We can also call as carrot rattle mosaic virus. I think so. So which is a virus where it does not code any code protein. The example is Umbra virus. So the cross production was otherwise called as pre-immunization, otherwise called as budwood grafting, otherwise called as super infection. So the answer is all the above. So these are all the other names of cross production. I hope you guys know cross production was first introduced by McKinney. Originally it is developed for tobacco mosaic virus, but now we are commercially exploiting for citrus tristeza virus. So size of tobacco necrosis satellite virus, I think sizes we might have discussed in earlier. Anyway, I will tell you once again. So generally the size of tobacco necrosis satellite virus is 17 nanometer. And tobacco necrosis satellite virus, there is a difference. Satellite means the smallest variant. Tobacco necrosis satellite means smallest virus, 17 nanometer. Tobacco necrosis virus is 26 nanometer it consists of. Okay, and the size of cauliflower mosaic virus is size having the range between 50 to 52. So 50 in between 50 to 50. So these are all the couple of important plant viruses and their sizes. Okay, satellite necrosis virus 17 nanometer, tobacco necrosis virus 26, brome mosaic virus 28, cowpea mosaic virus 28, tobacco ring spot virus 28, cucumber mosaic virus 30, Zika virus 30, tomato bushy stunt virus 32 to 35, alpha mosaic virus 30 to 57, cauliflower mosaic virus 50 to 52. Lettuce necrotic virus 60 nanometer, potato yellow dwarf virus 90 nanometer, Rio virus generally range between 65 to 70. This wound tumor, very important one, which consists of genome as double stranded RNA, I think so, right? So 70 nanometer, sugarcane mosaic virus 11 into 750, bean common mosaic virus 12 into 750, plumpox virus 12 into 760. And Badna virus generally ranges from 300 to 200. And tomato spotted wilt virus is 100 nanometer. Generally, in size it is a very largest 100 nanometer, spherical, and it actually like coiled kind of things will be there, but these are all a couple of information of plant viruses and their sizes. So these are all the couple of detection tests. Just now I told you we discussed about tetrazolium chloride. Like that, there are a couple of other detection tests also there. Anyway, sodium hydroxide test for kernel bunt of wheat and ergot, brain flotation technique wheat Tundu disease, Ouzo to test bacterial diseases for almost all bacterial diseases like bacterial blight of rice and other all kinds of bacterial disease. We do recommend Ouzo test. ELISA for especially viruses. What is ELISA? Enzyme linked immunosorbent assay, right? Tetrazolium chloride banana bunch top, just now we discussed it, right? Iodine test rice tungro virus, page polyacrylamide gel electrophoresis, or next one more thing is the SDS sodium didosyl sulfate polyacrylamide gel electrophoresis, I think so. So fluoroglucinol test. These are the various kinds of tests used for the identification of pathogen virides. Duppy technique, generally used for phytoplasma. Paraquat technique for post-harvest disease. These are all a couple of detection techniques, and the pathogen that is generally used to detect by using the test. The next one is size of cauliflower. I think we have discussed 50, in sometimes 50 or maybe 52 also. Okay, Prunus necrotic ring spot virus is transmitted by Prunus necrotic ring spot virus is transmitted by pollen. Okay, this is a very important one. And indicator plant used for citrus Tristeza virus. So generally in plant virology we can use indicator plant for the identification or for the easy research kind of things. That was actually the use of identification plant in plant virology was first thought by K.M. Smith probably in 1930s, I think so. So the later slowly the indicator plant will become famous. Generally the indicator plant used for plant viruses is goosefoot, Chinopodium amaranthicole, something like that, right? And here we can also have a couple of other indicator plant for depends on plant viruses. Generally, the indicator plant used for citrus Tristeza virus is a claxi lime. Okay, indicator plant for potato spindle tuber viroid is a tomato. Like that, indicator plants are there. I think we have already tabulated. I think so. So use of indicator plant in plant viral studies was actually thought by, just now I told you, K.M. Smith. K.M. Smith was the first person who introduced the use of indicator plants in plant virology. Bendy yellowwing mosaic virus is transmitted by, it is known to be transmitted by whiteflies, Bemisia tabaci. Smallest plant pathogen in the world, virus, viroid, vemelu and spiroplasma. The smallest known plant pathogen in the world is a viroid. Viroid is the smallest, very small than any other kinds of plant pathogens reported so far. And these are all a couple of indicator plants. Virus indicator plant is a goosefoot. The scientific name is Chinopodium amaranthicole. In 2021, they have asked the scientific name. Rather than giving the indicator plant, they have given the scientific name. So it is very important for us to identify or to remember the scientific name also. And citrus greening, sweet orange, the scientific name is Citrus sinensis. Citrus exocortosis, trifoliate orange, Citrus trifoliate. Citrus Tristeza virus, this was asked, I think so. Citrus Tristeza virus indicator plant. They have given the scientific name instead of giving the indicator plant as like Claxi lime or Mexican lime. They have given the scientific name of the indicator plant, that is Citrus arundifoliae. Okay, potato spindle tuber viroid, tomato. Solanum lycoperdum, Solanum berthali, right? Red rat of tea, fish poison bean, and phytoplasma, the indicator plant is periwinkle. The next one is which plant virus produce a large number of virus particles in the cytoplasm of infected cells. Generally, this potato virus produces a large number of virus particles in the cytoplasm. Okay, specific medium recommended for the production of monoclonal antibodies. There is a medium called HAT medium, hypoxanthine aminopterin thymidine, I think so. Actually forgot. I think it is more or less similar, like hypoxanthine aminopterin thymidine, I think so. It is the best medium. HAT medium is same. Okay, so this is the best medium used for the production of monoclonal antibodies. Okay, vaccination is derived from Latin word which means vacca means generally I think cow. So the meaning of vacca is cow. Inclusion bodies are otherwise called as elementary bodies, are also called as occlusion bodies. So the answer is A and B. Okay, inclusion bodies staining agent. What is the staining agent used for inclusion bodies? Azurea. CAMV, cauliflower mosaic virus producing inclusion bodies are known as. So cauliflower mosaic virus producing inclusion bodies are known as amorphous inclusion bodies. Anyway, we have another tabular column that shows what is inclusion bodies, what are all the different kinds of inclusion bodies that is produced by the pathogens. Okay, cauliflower mosaic virus produces amorphous inclusion bodies, whereas a pinwheel or cylindrical inclusion bodies are produced by 1/15th. Okay, produced by Poti virus, whereas bullet shaped inclusion bodies are produced by rhabdovirus. So I will show a couple of other things. So generally this one, tobacco mosaic virus produces X bodies, cauliflower mosaic virus produces amorphous inclusion bodies, Zika virus produces granular inclusion bodies, rhabdovirus produces bullet shaped inclusion bodies, whereas Poti virus produces pinwheel inclusion bodies. These are the couple of inclusion bodies that is produced by plant viruses. Bullet shape. bullet shaped, yeah, I think we have discussed, right? Bullet shaped inclusion bodies are produced by rhabdoviruses. We have already discussed the different kinds of inclusion bodies. That is also sometimes we do expect questions, right? So anyway, the next one is a test used for the identification of rice tumor virus. I think different identification tests also we have seen. So the test used for the identification of RTV is iodine test, right? Ouzo test is for bacteria generally, right? So the commonly infecting virus of orchids or orchids in worldwide, there is a virus called odontoglossum ring spot virus. Is the commonly and most commonly infected virus in the worldwide and India also. Okay, seed-borne viruses are transmitted through. Generally, seed-borne virus, which means they are transmitting. Generally, by means of vertical gene transfer, the seed-borne viruses are transmitted. Viruses are transmitted through vector, which is considered as virus transmitted through vectors are considered as. Just a minute. Virus transmitted through vectors, which is considered as okay. This is also vertical gene transfer only. Seed-borne viruses are transmitted through vertical gene transfer as well as viruses transmitted through vector, which is also considered as a vertical gene transfer. Okay, grape fan leaf virus is transmitted by grape fan leaf virus is best known to be transmitted by Ziphinema index. Nematode transmitting. Uh, okay, it is the question is supposed to be asked like nematodes which are involved in the transmission of plant viruses. Actually, the question was a little bit not well formed. Okay, so just change this question to what are the different kinds of nematode that act as a vectors in transmitting of plant viral diseases. So the answer is all the above, because this Ziphinema, Longidoros, Paralongidosus, these are all the very important nematode vector which act as a transmitting or vector for transmitting of plant viral diseases. So grape fan leaf virus, tobacco rattle virus, tobacco ring spot virus, P. early browning, these are all are known to be transmitted by nematodes. Ziphinema index, Trichodoros, Paratrichodoros, Ziphinema, Trichodoros species, Trichodoroid species. These are all the couple of species. Okay. RNA viruses RNA viruses generally multiply in cytoplasm. DNA viruses multiply in nucleus. Potato mop top virus is transmitted by, there is a virus called potato mop top virus which is known to be transmitted by fungus. We can also call it as furroviruses. What is furroviruses? Very simple. Viruses that are transmitting through fungi are known as furroviruses, which means the fungi is act as a vector in transmitting a plant viruses. Potato mop top virus transmitted through fungi, right? The next one is the coconut swollen shoot virus and a banana streak virus. Two important things are known to be transmitted by mealybug, Pseudococcus nijalensis. I repeat, cocoa swollen shoot and banana streak virus, both are known to be transmitted by mealybug, Pseudococcus nijalensis. Okay. Tomato spotted wilt viruses are transmitted by, tomato spotted wilt virus is generally transmitted by thrips tabaci. Bimodal transmission can be observed, and actually for the first time bimodal transmission can be observed in cauliflower mosaic virus. This question also we have discussed in the in between 1 to 50 questions, I think so. The bimodal transmission was first reported by Chaffant and Chaffman in 1962 in cauliflower mosaic virus. So this cauliflower mosaic virus is known to be transmitted by two means. One is sap transmission, second one is aphids transmission. Okay, so the groundnut bud necrosis virus is transmitted. This question was it's a very important one. This question was asked repeatedly many times, not in net but in other exams. Okay, whatever exam is, it remains exam, right? So groundnut bud necrosis is transmitted by thrips. And next one is the most sensitive technique used for the detection of plant pathogen. There is a technique called ELISA. This is the most sensitive technique used for the detection of plant viruses. Enzyme linked immunosorbent assay, right? The next one is most sensitive, ELISA only we have discussed, right? Enzyme used in ELISA. So enzyme used in ELISA, there are a couple of enzymes like horseradish peroxidase, alkaline phosphatase. I think they have given here, right? Both are used as ELISA enzymes. Substrate used in ELISA, para nitrophenyl phosphate. Sensitivity of ELISA. Next question: what is the sensitivity of ELISA? 1 to 10 nanogram per ml, not picogram, nanogram per ml. Okay. Promoter, which is a 35S transcriber belonging to the virus. So this 35S promoter is a very famous promoter which is used in gene transfer, that is actually originated from cauliflower mosaic virus. Plumppox virus is otherwise called as Sharka virus, not other things like Sharka. Okay. The protein subunits of tobacco mosaic virus is arranged in, they are actually arranged in a double helix. Okay, like this, this is TMV. This is how it is actually arranged, double helix structure in banana tomato, in tomato mosaic, sorry, in tobacco mosaic virus. So since there are a couple of questions were asked in ELISA, just have a look into what are the other different kinds of things that we must take into consideration, especially in the exam point of view. So the first one is the most sensitive technique used for the detection of viruses is ELISA. Enzymes used in ELISA, horseradish peroxidase and alkaline phosphatase. What is the substrate used in ELISA? Para-nitrophenyl phosphate or TMB, tetramethyl benzidine, T3, 3, 3, 5, 5 tetramethyl benzidine. Okay, so who coined the term ELISA? The term ELISA was coined by Eva Evangal and Peter Pellman 1917, but the technique this was given by Clark and Adams. Okay. Anyway, next one is a well plate or micro plate of ELISA was invented by. Actually, the plate we are using 96 wells, right? So the plate we are using is made up of Guy Takasa in 1951. Okay. So sensitivity of ELISA, how sensitive the ELISA is 1 to 10 nanogram per ml. The sensitivity of PCR is 1 to 10 picogram. You can remember PCR, picogram, ELISA, nanogram, like that. Okay. So what is the plate used in ELISA? So the plate actually composed of polystyrene plate. Okay. So most sensitive technique used for the detection of plant is same. Okay, number of wells present in ELISA generally it consists of 96 wells. Even PCR also consists of 96 wells only. So of course in ELISA variants are available 96, 72 and 384, 5300 and something plus. So there are various kinds of plates are available, but the most commonly used and standard plate consists of 96 wells. Okay, what is the pore size of ELISA? Each pore, we have 96 wells, right? Each well size, each pore size comprises of 0.4 microliter. It is supposed to be microliter, not micrometer. There is a typographical error. It's a microliter. Okay. Next one is the first virus code protein is fully genome sequenced in its amino acid. So for confusing us, they are asking in a different way. So first virus code protein is fully genome sequenced is tomato mosaic virus. Okay, virus which multiply in their respective vectors is known as propagative vectors. If they are multiplying inside the plant insect vector, it is known as propagative vectors. Okay, most of the plant viral transmitting vectors belonging to the order, majority of the plant transmitting viral diseases, sorry, the insects belonging to the order is homoptera. Okay, the number of protein subunits in tobacco mosaic virus are arranged in a helix per 3 terms. What is the number they are asking? 49 terms. It consists of 49. Okay, and the virus moves approximately 8 to 10 cells per day or per hour or per minute or per second. Actually virus is very small, so that it can only move 8 to 10 cells per day, entire day. Okay, infection of Gemini virus leads to, so generally just a minute, yeah, the infection of Gemini virus generally leads to what? It is generally leads to plate like inclusion of nucleus. Okay, so which of the following viroid is known to infect monocots and kill the entire plant? This is a very important question. There is a viroid called potato spindle tuber viroid. Sorry. Yeah, potato spindle tuber viroid only. Just a minute. This infection of Gemini virus that leads to hypertrophy in plant cell. Probably, so please change this. Infection of Gemini virus leads to hypertrophy in nucleus. Okay, and 143. The viroid is actually monocots is coconut kadan viroid. Okay, it's not potato spindle tuber viroid. So the infection of viroid that actually known as a monocot infecting viroid is coconut kadan viroid. Please change this 142 and 143. Okay. Cucumber mosaic virus is known to be transmitted by aphids and as well as sap transmission. Which of the following technique is used for reduction or reduction of eradication of viruses in a plant technique? So all the above techniques like chemotherapy, thermotherapy, meristem tip culture, all are used to eliminate the plant viruses. Which viruses have no vector? There is a virus called ILAR virus, Umbra virus, I think so. ILAR virus or carrot rattle mosaic virus or Umbra virus. So I think what we have discussed is which do not code protein. So that is also known virus which consists of no vectors. Luteoviruses are confined to. So luteoviruses are confined to only phloem. Okay. Most of the insect vector transmitting viral diseases have which type of mouth parts? This is an important one. So majority of the insect vector that is transmitting plant viral disease, they consist of piercing and sucking type of mouth parts. The matured virus particles is known as virion. Virion is a matured virus particle. So viride is what? Nucleic acid divide of protein code. Nucleoprotein means combination of nucleic acid and protein. We can call the virus as a nucleoprotein, which means consist of nucleic acid and protein code, like that. Okay, if the plant get infected with viruses, generally there is what? So generally the primary symptoms are like though reducing the chlorophyll, the plant leaves will become yellow in color, and options was actually reduced here. So the answer is actually all about decreased photosynthesis, increased respiration, and yellowing of leaves, and the option is all the above. And for further more information, students can refer this book, A Vision into Plant Pathology, a complete student version. For further doubts, guidance, and clarification, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.